In today's video, we're going to pick a question from my Microbiology IT Questions and Answers book. Now, my book has 450 plus questions and 450 plus answers to help guide you through each chapter as well as help you retain what you study. Now, if you need a guide to help you through your chapters, then check out my book. The link is in the description box. The question we will land on is, what are the three mechanisms of phosphorylation? Explain each one. Now, this question comes from the microbial metabolism chapter. Welcome to our deep dive into the world of phosphorylation. Phosphorylation is a critical process that drives many cellular activities. For example, energy production. In this video, we'll explore three key types of phosphorylations: substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. Are you ready? Let's get started. First, let's talk about substrate level phosphorylation. This type of phosphorylation occurs when a phosphate group is directly transferred from a high energy substrate to ADP, forming ATP. Now, this process typically occurs during glycolysis and the Krebs cycle. Let's see how it works. On the screen, you have a visual. We have our substrate, our high energy bond that connects the phosphate group. That high energy bond and that phosphate group connects to ADP, forming ATP, our energy currency. In glycolysis, there are two examples that I want to discuss that shows substrate level phosphorylation under the energy conserving stage. The first example here, it's 1,3-diphosphoglyceric acid. We have two molecules of that. Here you see that we have our phosphate group on each one with the high energy bond. When that bond is broken, the energy is released, giving two phosphate groups. The two phosphate groups will then attach to two ADPs, thus giving two ATPs. The molecule in the end, you'll receive two molecules of 3-phosphoglyceric acid. The second example under energy conserving stage is two molecules of phosphoenolpyruvic acids. We have the high energy bonds connected to the phosphate groups. The energy bond breaks, releasing the energy, and the two phosphate groups, they attach to two ADPs, thus giving our two ATPs, our energy. The molecule then molecules become pyruvic acids. You'll have two pyruvic acids in the end. In the mitochondrial matrix, we have this reaction, the succinyl-CoA reaction, where we have inorganic phosphates attaching to ADP to get ATP, as well as inorganic phosphate groups, phosphates attaching to GDP to get GTP, another energy source, resulting in the final molecule, succinic acid. Next up is oxidative phosphorylation, the powerhouse of cellular energy production. Now, this process occurs in the mitochondria, where electrons are transferred through a series of protein complexes known as electron transport chain. Now, the energy from these electrons is used to pump the protons across the inner mitochondrial membrane, creating a a protein, a proton gradient. Now, as these protons flow back into the mitochondrial matrix through ATP synthase that you see here in purple, the enzyme harnesses this energy to convert ADP into ATP. And this is how the majority of ATP is produced in aerobic organisms. Now, we specifically have here the cyclic photophosphorylation, where the electrons do go down the electron transport or go through the electron transport chain, in result giving ATP, but the electrons will then go back to chlorophyll. So that is the cyclic part of photophosphorylation. During the non-cyclic photophosphorylation, we do not see the electrons going back to chlorophyll, but they are used to help produce ATP. We have photosystem 2 and photosystem 1. And then in photosystem 1, we have the electrons going through the electron transport chain, and the electrons are used to produce NADPH. Here's an extra boost. If you are having trouble studying microbiology, check out my interactive slides where I provide you questions and it's up to you to choose the right answer. If you get the right answer, then you'll see on the next screen correct. But if you click the wrong answer, then you're going to see wrong. But you're able to go back to the original question and answer it. Now, if you find that this is something that you want and it will help you, go ahead and click the link in my description box. I'll have not only my Microbiology IT questions and answers book for you, but I'll also have interactive slides for each chapter that is and will be available for you. If you found this video helpful, don't forget to like, subscribe, and leave a comment below with any questions or topics you'll like me to discuss in the next video. Well, thanks for watching.